Okay, let's look at question 4. For question 4, right, I hope you can see that the phosphoglyceride is actually the normal default phospholipid that you have learned. The phosphosphingolipid is another type of phospholipid. Right, so for the phosphoglyceride, right, the normal structure of the phospholipid, the hydrocarbon tail shown here is a representation. It's not showing you the chemical structure, right? Yeah, and even though the box is given as a straight chain, but again, it's a representation, right? So there is really no evidence or no strong evidence to suggest that all these hydrocarbon tails are saturated, right? And they are chemically the same. I don't even think they are of this equal length, even though I've accepted that as an answer, right? So yeah, there are stronger evidences, more clear-cut differences, right, shown in the diagram. So as such, right, I am not accepting suggestion that the hydrocarbon tails are all saturated, but they are all identical chemically. All right, it's a pictorial representation, right? You don't really see the chemical structure. Okay, right. So that's why the differences are shown here that I will accept more readily. All right, the OH, well, you can't argue against that, right? OH on the phosphosphingolipid, which is not in the phosphoglyceride. The ester bond, right? Ester bond is given as this COC double bond O, right? This is the ester bond, right? You don't see that on the phosphosphingolipid. Right, some of you mentioned that this on the phosphosphingolipid, this is a amide bond or peptide bond. Even some of you mentioned that. Okay, I accept that as a difference as well compared to the ester bond on the phosphoglyceride. Okay, presence of nitrogen, very clear cut. Right, so all these are more correct, right, compared to some assumption that the hydrocarbon tails are the same. So again, that's why these are the more or better answers, right, that I will accept more readily. Then the CC double bond that you can see here, I suppose that's the reason why some of you, or most of you actually, mentioned that this hydrocarbon tail in green is unsaturated and the rest are saturated. Right, but again, we wouldn't know whether within the hydrocarbon tail themselves, right, there are any double bonds. Also, you can say that there is this additional double bond besides whatever that may be found, right, in the hydrocarbon tail itself, okay, which is not present in the phosphoglyceride. Okay, and if you are a bit more familiar with the structure and the formation of phospholipids, right, especially the phosphoglyceride, the default structure of the phospholipid, I hope you can see that yes, this is glycerol, right, with the 3 carbon. The O, 1, 2, 3, right, these 3 O's, I follow my cursor, 1, 2, 3, these 3 O's belong to glycerol, right, because in the formation of the ester bonds and the phosphoester bond, the glycerol loses H, right, for each bond that it forms. Yeah, so these 1, 2, 3 O's are belonging to glycerol, right? So these 2 hydrocarbon tail with this C double bond carbonyl group, right? So this carbonyl group is the remnant, the remaining part of the carboxylic acid group, right? C double bond O, OH, right? The OH is lost in the formation of the ester bond, right? So these 2 hydrocarbon tail with the carbonyl groups, right, or what used to be the carboxylic acid group, they are both fatty acids, right? So this is again the normal structure of the phospholipid that you learned, right? 1 glycerol, 2 fatty acids, and 1 phosphate group. But for the phosphosphingolipid, right, so you can see that the one in black with the carbonyl, this is a fatty acid, right? But the other hydrocarbon tail is not a fatty acid. It doesn't carry the carbonyl group or the remnant of the carboxylic acid group, right? So this is not a fatty acid, right? So only a couple of you managed to see that and gave that as a difference, right? 2 fatty acids versus 1, which I've accepted, all right? And for the glycerol part, this is really not glycerol, right? For the part on phosphosphingolipid, because again, right, remember glycerol 1, 2, 3 O's, right, belongs to glycerol. Here that you have the OH here, you have the O here, which used to be OH, but there's no OH here. This is N, right? So glycerol doesn't contain N, right? Yeah. So this 3-carbon structure, right, is not really glycerol. Yeah, and I don't think anybody managed to see that, or at least not in the answers that you have given. Okay, and also, right, ester bond, right, is COC with the double bond O, right? This is CNC with the double bond O, this N, right? So this is not a ester bond, right? So quite a few of you mentioned that, oh, there's one ester bond in the phosphosphingolipid. I assume you're referring to this, but this is not an ester bond. Yeah. Okay. So again, anyway, these are the differences that I have accepted, and there are a few more that I did not include, right? But really not identical hydrocarbon tail versus different hydrocarbon tail, all right? Or that they are saturated versus unsaturated. Again, because we don't see the chemical structure of the hydrocarbon tail. Okay, then for part B, right? This one is a bit more straightforward and quite well done, actually. Right, the rough region, as you can see, right, has got more cholesterol. Those in yellows are cholesterol, not proteins. Proteins are much larger than this, right? So the molecule that's embedded between hydrocarbon tails of phospholipid, the only thing you learn really is cholesterol. Okay, so these yellow stuff are cholesterol. There are more of them, right, in the rough region versus the non-rough region, right? The fatty acid tails or the hydrocarbon chains are all straight and therefore saturated versus saturated and unsaturated. So a mixture, right, for the non-rough region, only saturated in the rough region. Okay, then the packing as well, right, much more closely packed in the rough region than the non-rough region, right, packing of the phospholipids, that is, yeah. Again, quite well done, so let's move on to part 2, right? So part 2, the question is on permeability, right? Permeability, not fluidity, right? So, I don't know whether you remember in CA2 when we went through one of the questions that I marked. Yeah, which is also, there was a question also on fluidity versus permeability, right? And you mixed up in CA2, right? And in the 2 LTs, I was trying to explain the differences between fluidity and permeability, right? They are related, but they are not the same. But here, again, right, when the question is about permeability, I keep seeing people talking about fluidity, right? How the phospholipids can move versus cannot move laterally. All those are not talking about permeability, right? So can we once and for all just remember that permeability refers to how easy it is for substances to pass through the membrane or diffuse through the membrane, right? Versus fluidity is about how easy it is for phospholipids and proteins to move laterally, right, in the membrane, how flexible the membrane is, right? So permeability and fluidity are describing different things and therefore are different concepts. Okay, they have relations, right, but they are not the same. Yeah, so when the question is asking you to comment on the permeability, then there should be focus on how easy it is, right, for substances to diffuse through the membrane. So in this case, for the lipid rough region, the phospholipids are closely packed, right, with very little gaps or pores between them, and also the cholesterol blocks any potential pores, right, any gaps, right, to to prevent leakage, which is one of the functions of cholesterol, by the way, by preventing leakage across the membrane. And so because of this, right, close packing, no pores, no gaps, and the cholesterol as well, right, then the lipid raft has low permeability because substances would not be able to diffuse through the lipid raft that easily, right, because of the lack of gaps. Not fluidity, not how the membrane is rigid, how the phospholipids cannot move, right? All those are fluidity, not permeability. Okay, then for part C, right, is on phagocytosis, right? So phagocytosis involves the extension of the pseudopodia. The word is extension, right, of the pseudopodia to surround the pathogen, right, and put the pathogen in a phagosome and engulf the pathogen, right, into the macrophage or the neutrophil or any other phagocytes. So it's extension, right, of pseudopodia, which requires the membrane, the cell surface membrane especially, to be able to bend, to change shape, right? So that requires high membrane fluidity. This question is on fluidity then, right? Not permeability anymore. So the region of the membrane that has high fluidity, of course, is the long raft region. You can see that the phospholipids are further apart and there are kinks, right? The unsaturated hydrocarbon tails, right? So that makes the membrane more fluid. Okay, so with high fluidity, the membrane is able to extend out, right, change shape, and forms the pseudopodia. And the pseudopodia also is more able to fuse together, right? After the two pseudopodia have surrounded the pathogen, right, to fuse together to form the phagosome. Right? All this requires fluidity to be high. Okay, so it's not about permeability because pathogens are big, right? They are not some small molecules or ions, right? Pathogens don't diffuse across the membrane, right? No matter how porous or permeable the membrane is. Yeah, so this is not about how you should be looking for a lower permeability so the pathogens don't escape, which is what so many of you have written, yeah? But pathogens cannot escape anyway, right? Because they are too large to diffuse across the membrane, regardless of how permeable the membrane is. So this is not about permeability for those who have chosen the raft region and saying that it is less permeable, right, to trap the pathogen, yeah? Okay. Then there are a few more misconceptions on the role of cholesterol. Okay, I'm not going to go through it here because it's pretty basic and not many of you have made those misconceptions. So I'll leave you to read a bit of the comments or to study cholesterol on your own. If not, please look for your own tutors for clarification on the basics of the functions of cholesterol. Okay, so that is question 4.